Welcome friends and welcome to my channel Sequential Biology. We are talking about viruses and today we will discuss about bacteriophage and how many types of bacteriophage can be seen. Okay. Now the bacteriophages are viruses that infect and replicate within bacteria and destroys the bacterial cell, and for this reason the bacteriophage are known as beneficial viruses. Then bacteriophage are nothing but this is a type of viruses. Since this is a virus, so bacteriophage must contain capsid layer and their genetic material. They are also called as phages. Bacteriophages are also known as phages, or bacteriophages are called as bacteria eater since they destroy bacteria when they attack bacteria, and so the bacteriophage are known as bacteria eater. Bacteriophage can be found everywhere. They contain DNA or RNA in their genome. In any living organism, both these two types of nucleic acid can be found, both DNA and RNA can be found, but since this is a virus, either DNA or RNA can be found in their genome, and which is encapsulated by protein coat. And you know, in any viruses the protein coat or the capsid are present, and inside this capsid their genetic material is present, and this genetic material may be either DNA or RNA. So bacteriophage may be DNA containing bacteriophage, or bacteriophage may be RNA containing bacteriophage. And since this is a virus, so this nucleic acid are covered by protein coat, and which is also known as capsid. They also infect archaea. They not only infect eubacteria but they also infect archaea. And we know the Monera, the kingdom Monera, are divisible into two broad categories. One is archaea, and usually the archaea are present in extreme environment. Okay, and eubacteria are present everywhere, and archaea are present in extreme environment. Eubacteria present in everywhere, and so the bacteriophage not only attack eubacteria but also they can attack archaea. William Todd discovered bacteriophage. Then who first discovered bacteriophage? William Todd in 1915. He discovered bacteriophage. The number of genes in bacteriophage may be 4 to several hundred, and the 4 genes are present in MS2 bacteriophage. Only 4 genes are present in MS2 bacteriophage. D. Harrelly coined the term bacteriophage. Then William Todd first discovered bacteriophage, and D. Harrelly coined the term bacteriophage in 1917. The most common and the diverse entity in biosphere. Different type of bacteriophage can be seen in biosphere. Biosphere means the region of earth where life can be seen. So this is the most common and the diverse entity in biosphere and found wherever bacteria exist. It is estimated that there are more than 10 to the power 13 bacteriophage on the planet. 10 to the power 13 bacteriophage are present in our planet. Bactericidal activity, that means they kill bacteria, so this is called as bactericidal activity. Bactericidal activity of bacteriophage was first observed in 1896 by E.H. Hankin in the water of river Ganges. Bacteriophage in river Ganges, the bacteriophage are present and which could kill cholera bacteria, and for this reason the Gangetic water is known as pure water. Okay, so in 1896 E.H. Hankin in the water of river Ganges, the bactericidal activity of the bacteriophage was first observed by E.H. Hankin in the water of river Ganges, and since this bacteriophage attack different type of bacteria in water of river Ganges, and for this reason the water of Ganges is very much pure. Okay, and which could kill the cholera bacteria. Now the bacteriophage are two broad categories, the T odd phage and T even phage. What is odd number? Means 1, 3, 5, these are odd number, and even number means 2, 4, 6 are even number, and which are divisible by 2. They this number are known as even. Okay, now the virulent phages kill their host. Virulent phages kill their host, and these phages are designated as T1, T2, T3, T4, T5, T6 and T7. Virulent phage means the harmful phages and which kill bacteria, they are known as virulent phages. So virulent phages kill their host, and these phages are designated as T1, T2, T3, T4, T5, T6 and T7. The most important virulent phages are T7 and T3 and T7 phages. The T refers to tail of these phages. Sometimes we confuse that why we use this T term. Then this T term is nothing but this is the tail of these phages. You know the bacteriophage has tail. Bacteriophage, this is a capsid layer, and also the bacteriophage has tail. So this T means the tail of these phages they contain. There are 3 even numbered phages. So the T even number phages T4, T2 and T6, these are T even number phages, and the T odd number phages are T1, T3, T5 and T7 that infect the E. coli, so they are called as coliphages, right? So these 4 odd numbered phages T1, T3, T5 and the T7 that infect E. coli or Escherichia coli, this is a type, this is a bacteria, and for this reason these T odd numbered phages are known as coliphages. Since they attack E. coli, so they are known as coliphages. Bacteriophages T2, T4 and T6 are closely related viruses. Now, except T3 and T7, all other phages are T series and have a tadpole shape with a hexagonal head and long tail. So this hexagonal head and the long tail cannot seen in case of T3 and the T7, but all other phages T1, T2, T4, T5, T6, so these all phages contains a hexagonal head and long tail. We will discuss their structure in next classes. Now the most important phages are T4, T2, lambda phage, M13 phage, 5x 174 bacteriophage. There are different type of bacteriophage, and here we will only discuss about important phages. T4 phage is only lytic cycle. T4 phage shows only lytic cycle. What is this lytic cycle? When any phage infect any bacteria, suppose this is a bacteriophage and this infect this bacteria, and this bacteriophage or virus replicate inside their host and they kill this host cell and rupture this host cell and come outside by rupturing of this host cell. And the viruses which shows this pattern of life cycle, they are known as lytic phages. So T4 is only lytic cycle, but no lysogenic cycle can be seen in T4 phages. What is this lysogenic cycle? When virus penetrate their genetic material into bacteria, and their genetic material will be incorporated with the bacterial genome, with their host genome, and the viruses which shows this type of reproductive strategy are known as lysogenic viruses. Then the T4 phages are only shows lytic cycle, that is when they attack any bacteria, then they kill this bacteria, they rupture their cell, and for this reason they show only lytic cycle, but no lysogenic cycle can be seen in T4 phage. And this T4 phage infect E. coli and most extensively studied bacteriophage. T4 phage is most extensively studied bacteriophage, and they have linear double-stranded DNA. That means their DNA looks like linear structure, and this DNA is double-stranded linear structure. Next, T2 phage. T2 phage is famous Hershey and Chase experiment to prove that the DNA is genetic material done with the T2 phage infecting E. coli. Then T2 phage also infect this E. coli, and this T2 phage was used by Hershey and Chase in their blender experiment. This is also linear double-stranded DNA like T4 phages. The T2 bacteriophage also have linear DNA. This DNA is not circular. DNA may be two type. The DNA may be circular, or this may be linear. Then this T2 phage genetic material is linear double-stranded DNA, and this undergo lytic cycle. Also this T2 phage undergo lytic cycle. And the next bacteriophage is lambda phage. This lambda phage also infect the coliphages and used as vector in biotechnology, and their genome is used in biotechnology, and their genome is used as cloning vector and undergoes both lytic and lysogenic cycle. Then this lambda phage undergoes both lytic and lysogenic cycle. Different type of proteins, CRO protein, C1 protein control this lytic and lysogenic cycle, and this lytic and lysogenic cycle will be choiced according to their needs. Then this lambda phage shows lytic and lysogenic cycle. Next, a bacteriophage is M13 bacteriophage, which infect E. coli, and it has circular single-stranded DNA. So this has a circular DNA, but this DNA is single stranded. Remember it. This is an exception. Usually DNA is double stranded, but in M13 phage we can see a circular single stranded DNA. And another bacteriophage which also possesses single stranded DNA is X174 bacteriophage. They have also circular single stranded DNA like M13 phages. It was first genome to be sequenced, so their genome first sequenced, okay, and also the first genome to be assembled in vitro. So in vitro means outside of body of any organism. That means in test tube. In vitro means outside of body of organism. And then the 5X174 bacteriophage's genome to be assembled in vitro, and the Sinkheimer discovered single stranded DNA in 5X174 bacteriophage. And the next is cyanophage. Cyanophage, these are also the viruses which infect blue-green algae. They attack blue-green algae or the cyanobacteria. Structure of cyanophage is similar to bacteriophage. An example of this cyanophage is LPP1, called so as it attack Lyngbya and the Phoridium and the Plectonema. So this type of blue-green algae, these are example of blue-green algae, Lyngbya, Phoridium and the Plectonema. These are blue-green algae, and this cyanophage attack these blue-green algae, and LPP1 is an example of this cyanophage. Then bacteriophage are usually two types, the temperate phage and virulent phage. The temperate phage does not cause disruptive infection, but virulent phage actually follow lytic cycle. And the temperate phage instead phage DNA incorporated into bacterial DNA and replicate, so they are lysogenic phage. The temperate phage are lysogenic phage. That means when they infect any bacteria, then they insert their genome into this bacteria and they will form a prophage. Suppose this is a viral genome or the bacteriophage genome, and when this bacteriophage genome incorporated into bacterial DNA, this is bacterial DNA, suppose this is bacterial DNA and this is phage DNA, this is phage DNA, and this when this phage DNA incorporated into this bacterial DNA, then this is called as the prophage. So they reproduce via formation of this prophage, and their phage DNA will be incorporated into the bacterial DNA, and for this reason they show the lysogenic phase. Okay, though temperate phage show lysogenic pattern in their life cycle, and also sometimes become virulent. So they may be sometimes become virulent. Sometimes they can rupture the bacterial cell and cause the lysis of this bacterial cell. Example of this temperate phage is lambda phage. The next virulent phage follow lytic cycle. They rupture bacterial cell, and for this reason they follow the lytic cycle, and their life cycle pattern is lytic cycle, and causing bacterial infection and eventually destruction and death of bacterial cell. Example T4 phage. This is the most extensive studied phage, the T4 phage. We have seen that the which are T odd phage and which are T even phage, and who are virulent phage and who are non-virulent or the lytic phage. And also we have talked about different important bacteriophage, T4 phage, T2 phage, lambda phage, M13 phage, 5X174 bacteriophage, and also example of cyanophage. And also we have seen that who are temperate phage and who are virulent phage. So thank you, and that's it.